Hi, I'm Gbola Amusa, and it's my pleasure to introduce our next guest from REGENXBIO. We have Curran Simpson, who's Chief Operations and Technology Officer. The format for this session is a roughly 40-minute presentation followed by fireside chat, so altogether it's 40 minutes. If you have any questions during the session, feel free to type them into the question box at the bottom, and I'll do my best to ask them on your behalf. Curran, thank you for joining us virtually today, so please begin the presentation.
Okay, thank you, and thank you for everyone for attending. I just have a short video. I know some of you may have had the chance historically to visit REGENXBIO, but for those of you that haven't, I thought I'd take you for just a quick tour of our labs where we have 125 plus people focused on developing our pipeline, focused on CMC-based activities. And our pipeline spans, as many of you know, from large indications to ultra-rare indications across multiple targets. So the Tech Ops team has been working tirelessly through COVID to continue the development and scale-up methodology to further advance our programs, especially as we begin the process of preparing to open up our new internal manufacturing facility in Rockville, Maryland. The improvements the team is making will have dramatic increases in the ability to serve patients, the amount of vector we can produce per batch, and ultimately the reduction of cost of goods. And we have exciting new technology being developed in our downstream team to better purify and enhance our product profile in gene therapy, which is a real focus in the area. So with this slide, I'll just walk you through some very sort of all the way back to 2010, where if you look at the inflection around 2016, that's when REGENXBIO began to bring a lot of internal capability in CMC on board in Rockville, starting with our first hires around that time. Up until that point, we had been operating largely with adherent cell culture processes, very small batches to supply our first clinical programs in what we called manufacturing 1.0. That process used an adherent HEK 293 cell line. And again, that was used to make our first clinical batches. As we headed into 2016, we could see that the future really required us to scale up dramatically from where we were. And this, of course, is a focus of the entire gene therapy industry, especially those focused on AAV, to a period of time where I call it manufacturing 2.x, 2016 to 2020. We adapted the adherent HEK 293 cell line to become a suspension cell line and also modified our upstream process to be bioreactor based. Along the way, really significantly expanding our capability to produce more material and to raise productivity of the cell lines as well. So as we think about that, and as we think about the future, it takes us to now, which is an extremely exciting time for the company as we expand into our corporate headquarters, which I'll show you will contain a 2,000-liter scale manufacturing facility. And as part of that scale up, we are optimizing our current 1,000-liter process to take us further into the 2,000-liter range. And as you see with the announcements for indications that we're exploring that are targeting the liver, for example, the vector requirements are going up substantially from our other programs. And in lockstep with that, we are enhancing our CMC capability to reach that scale. This picture, I think, says it all. If you look at 2017, moving out of the adherent cell culture process, you see what are called hyperstacks on the left, a lot of hands-on manual labor required, actually quite a robust process, as it turned out. The product profile, everything associated with adherent cell culture processing was acceptable for early clinical development, but the problem we faced was how do we scale this? How do we get to larger batch sizes, more in line with what you've seen the emergence over time in the biologics field? So flash forward to 2021, what you see on the right is Yes, we do still have people in the facility, of course, but it is highly automated. It's utilizing much more modern equipment, single-use technology, bioreactors, automated chromatography skids, all that can scale actually to 2,000 liters and beyond if necessary. So a significant change in just a few years for REGENXBIO and of course the industry as a whole. What does that result in that's tangible and important for CMC is the number of patients we can serve per batch over that period of time has increased almost 100-fold from where we started. So very, very significant improvements in productivity and a commensurate decrease in cost of goods. Now, we can all argue, I think, going back to 2016, relative to biologics, gene therapy manufacturing is still in its infancy. There's a long way to go on further yield enhancement, further reduction in cost of goods. But if the progress we've made in the last 3 to 4 years is indicative of what we think will happen in the next 5 years, we're going to get there and continue to excel at moving the industry more towards what you've seen historically, again, in biologics. It's easy to get focused on 2,000-liter capacity bioreactors and forget about the entire supply chain when you think about manufacturing. And so I just want to reinforce about our new manufacturing facility, our ability to scale to 2,000 liters, and the productivity improvements we've made. We've also spent an enormous amount of time securing all aspects of the supply chain, because if the ultimate goal is to be realized, which is to deliver drug to patients, all of these have to be in sync and none of the links in the chain can be broken. So focusing on raw materials, which has been a real challenge during COVID to access raw materials and acceptable inventories. Bulk drug substance we've talked about. Finished drug product is critical, not just the ability to fill drug product, but the ability to develop devices, the ability to meet the analytical requirements in your quality testing, and then all the way through to shipping, labeling, and packaging. Every one of these aspects has to be considered, and a company to be fully integrated has to excel in all of these capabilities or identify a viable partner to support them with that in order to make the whole thing work. And this is something we've done really well with over the last few years. I do want to just show briefly a 3D rendering of our manufacturing facility. This is located in Rockville, Maryland. And our corporate headquarters is in the same building. The process development group is on the 4th floor. And on the 5th floor, we have manufacturing with QC labs, as you can see, with material support, warehousing on that 5th floor of manufacturing. A fill finish suite, which is critical to do direct fills from our bioreactors. And then most impressively, 2 independent manufacturing trains at 2,000 liter scale, bulk drug substance train A and bulk drug substance train B. So we've packed an enormous amount of capability into a fairly small footprint, but which will suit us beautifully for launching new products, validating the final steps prior to filing BLA will be done in this facility for certain products. And as we'll talk about over the next 40 minutes, securing capacity for REGENXBIO for many years to come. So very, very important investment. And over the last 2 years, I wanted to give you a 30 to 40 second time lapse of the building coming together. So you'll see the building being fitted on the outside. And once that was closed up late last year, the internals were built, and in a very exciting way, the labs will be open probably in the next 6 to 8 weeks, where we'll be consolidating from 5 different buildings into one, which will be excellent for the team. And second, the manufacturing facility in late this year will be in commissioning and validation. to start our first GMP lots in Q2 22. So very, very near term, while we also enjoy the benefit of the partnership we have with Fuji in Texas, who's been our manufacturing partner for the last 5 years. We're building a really significant manufacturing network that can flexibly meet all of our needs. I've talked about process and I've talked about the plant. I did want to just have one final slide to discuss the staff. We've been able to attract really, really terrific, experienced professionals from all over the world, really, in our gene therapy CMC team. On average, the leadership team has 20 plus years in biopharma. We've collectively submitted 66 INDs. We've got 26 BLA or NDA submissions between us, and we've been part of licensing 23 commercial products. And in gene therapy, where development can move very fast, licensure can happen very early relative to some of the development cycles you see for biologics. You need to have that experience on hand early, because if there are issues early on that aren't recognized, it's very difficult to catch up. And so we treat every single manufacturing batch that we produce as if it was part of a pivotal study, because it could be. And that's why we wanted to bring in a team as seasoned as we did. The excitement of gene therapy made it easy to attract them, and I'm thrilled to be part of it. That is the end of my presentation. I hope that was informative, and well, I look forward to questions.
Thank you, Curran. It was more than informative. We even got time lapse. So well done on that production. So I just wanted to start by orienting the audience a little bit to what might be unique about REGENXBIO's platform as relates to manufacturing and the pipeline, the portfolio. So can you discuss the differences here? First of all, REGENX has a broad pipeline. So what does that mean for what your job has to accomplish?
And you'll hear this a lot from different sponsor companies. We do run what I would call a true platform. And what I mean by that is if you look at product A versus product B and C, 95% of the materials used in that process and equipment would be the same. So we're changing out just a few different components of the process between programs, which is how with 100 people you can leverage 7 different programs. So I think that was a really important early decision we made, was to standardize, not make each program a custom process, which other companies have done, but to really focus and make a platform approach to this. The platform gets tested, right? As you go into new indications where very high vector requirements may be present, you have to adapt that platform, which we are, to go to the higher scales that I've described. Scaling up a transfection-based process is not trivial. Everyone that is working on this would probably say the same thing. That's why we've brought in to the company engineers that have been used to working at 20,000 liter scale, who have really, really good skill sets around how to scale processes up. And that's why when you look at productivity at 50 liters and 500 liters, 1,000 liters, and ultimately 2,000 liters, you're going to see linearity in the amount of productivity. So we don't see yields reduced at higher scales, which obviously goes against the notion of scaling up. We see them sustained along that way. And that's by, good modeling of the process, good understanding of the equipment, and the know-how to how to scale processes up, which is really a core strength of ours.
Got it. And you talked a little bit about the team and who you have, and that seems to be super, super important in the space, with some companies talking about how difficult it is to recruit right now. So can you talk about what percentage of your overall organization in terms of people is dedicated to manufacturing and support functions? And then secondly, whether your local recruitment is easier than, let's say, in other regions of the country?
So the proportion of, so if the CMC team roughly is 125 now, it's growing to about 170 by year end. The additional growth will be primarily manufacturing associates that are being recruited for the new manufacturing facility. But most, if not all of the others are focused on process development, analytical development, quality control, manufacturing. Within the process development group itself, I would say it's around 60 of those FTEs focused on standard PD activities, upstream, downstream, analytical, things like that. But we do have a sizable quality organization because we have responsibility for product being made at our CMOs as well. So testing and release. And so a full quality unit is already in place. And we do a lot of the GMP testing internally already within our current facilities. The second question?
Oh, just about local recruitment.
Yeah, this is a terrific part of the country to recruit in, to be frank. We have a fair amount of biotech in the area, and that's growing. We have places like NIH, FDA, you name it, within reach. And so for recruitment, I actually think Maryland is ideal. I've been here for almost 15 years, and we're all pretty well networked. And I'd say the recruitment atmosphere, the number of students emerging locally from University of Maryland and other local universities, Johns Hopkins, provides a rich pipeline of young employees looking for their first job. And we have a very, very strong network. And I think also REGENXBIO presents a super interesting place for people. Maybe they've been working in biologics for 7 years and they're looking for something new. There's a real attraction to gene therapy for obvious reasons. You know, if you can't get excited about the opportunity to make a child who has a terrible prognosis well, I don't know what drives you to come to work. So that on its own just really helps us attract people. There's a super meaningful cause that people are associated with in our industry.
And part of the great presentation, you had those cost curves or curves showing the efficiency of your processes improving. So could you talk about what the main drivers are a little bit more? Was there, for example, one or two things that were most important as factors?
Well, I think it's two. Yeah, it really is two key areas, and the other ones will emerge as we get our hands around the first two. The amount of vector produced in the upstream process drives the cost of goods, you know, immeasurably. So we are really, really focused on that. We probably have, in terms of cell line development, upstream development, 30 people, say, focused just on that. How to get better expression, what's the best cell line to use, and how can we do that? Because if you have a good product profile upstream and a high yield upstream, the rest of it becomes quite a bit easier to do. Having said that, one of our board members pointed out to me rightfully that you lose as much in fill finish many times as you lose in your whole downstream process. Just small volumetric, keep in mind our bulks right now in gene therapy could be as little as 80 milliliters. So you think about, you're starting with a 500 liter reactor and your final bulk is 80 milliliters. If you take a sample, that's 5 milliliters. What percentage have you just lost? So we have to really focus on the upstream yields, and we have to really optimize the downstream yields and clever sampling of the product to get the best we can in terms of overall yield.
And with each process improvement, and you know, there's more to come, as you mentioned. Are you able to have big gains on cost, time, and volumes? Are all of those considerations possible?
Yes. So there are some differences. Again, people are so seasoned in biologics, I often refer to that, but the upstream process for a bioreactor in biologics can be somewhere between normally 10 and 14 days. So that becomes, in effect, your bottleneck and why people are focused on the capacity in the largest bioreactor. That's your production reactor. And that kind of sets the tone for your batch times, which is the amount of time you consume in the plant, which relates to cost of goods directly. In gene therapy, the upstream process tends to be much faster, maybe a third to a half of that. And so now the bottleneck is somewhere else. It could be the material flow through the plant that's constraining you, buffer prep, media prep, or perhaps downstream. Some of the unit operations that originally were used for gene therapy still are in some cases, like ultracentrifugation is a very, very difficult process to scale, and could consume on its own just a day of manufacturing for that single unit operation. So we're trying to work with scalable unit operations that can be done in less than 6 hours, for example. So I do think it's very instructive to step back and take off our biologics lens at times and reevaluate gene therapy, because I think there's a throughput model for gene therapy that's going to be different. And the sooner we seize on that and actually adapt to it, the faster you're going to see higher levels of productivity and therefore lower cost of goods.
Would you discuss any innovations that are on the horizon that can further reduce or circumvent some of the key cost drivers? And could you start with ones that are on the horizon and then ones that you wish were on the horizon as well?
I think on the horizon, certainly there are alternate cell lines that people are looking at. So people talk about HEK293 transfection processes. Other people are looking at other cell lines, HeLa, baculovirus. I think you have to keep your mind open on all of those near-term technologies, even sometimes by program, as to which one is the most productive. And what we're finding is that answer isn't always the same for every serotype. But those cell lines are ready to go, could be utilized now, and in some cases can afford significant breakthroughs in expression yields, for example. A little bit longer term for things like HEK293 would be the production of what are called producer cell lines that can operate free of plasmid, and therefore you avoid a large amount of the transfection process. I don't think that's a near-term solution, but it's fairly near-term. Next couple of years, I think you'll see those producer cell lines implemented more broadly, and you'll see the dependence on plasmid supply potentially coming down somewhat. Still a lot to be worked out. The downstream chromatography area, there are near-term innovations, which we are part of. One of the focuses is separation of empty and full capsid, which is very, very difficult to do without a yield hit that is very detrimental. And that's an area where I think every producer is working very hard to optimize. We've made really significant progress there, but much more work to do. But it all has to be scalable. We have to avoid things that worked well in an academic setting, but do not work well at 2,000 liter scale. We have to get those out of the system, if you will.
Understood. So I guess that's both things that are on the horizon and you wish to. Okay. On the issue on full to empty capsids, I heard earlier today that sometimes it's full, but with the wrong material. Is that a real issue across?
It can be. I think as we all get better at knowing our products, again, gene therapy being an emerging field, we understood 5 years ago, empty, partial, and full. That was, something you would run a ultracentrifugation run and understand your product profile that way. Now people are doing mass spectrometry of both empty, partial, and full capsids and looking at the what's inside basically and seeing differences in the way the capsid is assembled. And so knowing that and knowing how that may change between manufacturing batches, for example, is very, very important. And knowing how to separate out these product-related impurities will become even more important as we move forward too. So that's been something that I think we've been focused on literally 5 years back, knowing that we have to get more towards full capsid and that the product profile is consistent batch after batch after batch, which really predicates an understanding of what the plasmids are doing, how can we optimize them for better packaging. That's a real exciting field that I think is also emerging.
Got it. So you've got a new facility, so I just am interested in some of the specs around it. So could you talk about capacity limits, scalability, versatility?
Sure, and that's a good point right out of the gate, is that the facility, I describe it as two 2,000-liter trains. We lead up to that with a 500-liter vessel ahead of the 2,000 and then a 50-liter vessel ahead of that. We can run in an extremely flexible manner because not all indications require that scale. So what might work in one indication and be necessary might not be the case in the other scenario. So what you're trying to think about is what's the optimal scale to operate at, such that I get enough manufacturing batches so that I can easily file my BLA. Because if you only make one batch and that's what you have to file on, you're going to have to make a lot of post-approval commitments to supply more data. So going to large scale wouldn't make sense in that instance. And so we can operate in our facility at 500-liter scale comfortably. We can operate by combining 500 liters to make 1,000-liter scale, and as high as 2K, depending on the program and depending on the need. That flexibility was built in on purpose, knowing our different indications might require different scales of operation. As well, the fill finish line has to be able to accommodate multiple vial configurations. So we're trying to be as flexible there so that we don't custom build a particular fill line with one vial size being acceptable and then not have that work for 80% of our indications. We want to try to keep that as consistent as possible and as flexible as possible for different configurations. The only one that I would say is very particular to the indication is the development of the device. And so for the ocular indications, we have very specific custom devices developed, whereas for IV administration, it's a traditional vial configuration. But in the manufacturing setting, the other piece of this that's important is having two segregated suites allows us to run two different programs simultaneously. That's really, really important. And so we can avoid having to go back and forth with campaigns of different products in the same suite, really expensive long changeovers that cost time and money. So that flexibility will really pay off in terms of our ability to, on one side of the facility, advance an early stage clinical program, and maybe on the other side of the facility, we're making a product that's going through the last stages of process validation.
So what else does the new facility have that, let's say, facilities that your peers built years ago might not have?
Well, it takes full advantage of the single use. Now, that's been around for a long time, but most people miss one of the biggest benefits of single use. We started building the manufacturing plant 18 months back, and we're going to have it in GMP operations in about 5 or 6 months, or actually more like 8 months. The reason we can do that, I don't have to validate 100 CIP lines or 100 SIP lines. That was the way most traditional manufacturing plants were built even 5 years ago. Everything now is single use, so all of that validation is not necessary. We have a single use equipment, bioreactors, and the buffer prep areas. So the speed at which you can stand up a new manufacturing plant is light years faster than it used to be. And the speed at which you can change over from one product to the next is the same. So I feel like that's an advantage. Now, that's an advantage that everybody should be taking stock in now as they build their own facilities. But building it in initially as that flexible will have long-term benefits for us. And I'd say some of the other things we've done, we've designed the facility to be able to run multiple types of cell lines. So it's not dedicated to, for example, HEK 293. We could run a different cell line in there in the way that we've designed the bioreactors. So yeah, I think just generally that plus what you really have to value the most, in my opinion, is being one floor away from our process development group. Is you cannot measure the benefit that that gives you. Having the knowledge transfer, being one floor away to your manufacturing staff from the process development staff is a huge benefit for any company.
Understood. And so what sort of guidance or conversations have you had with the FDA on the new facility?
So we have done a formal design review of the facility, and we got some comments regarding that design review, fairly minor. So that's where we are. And we've also employed various QP reviews to make sure the EU perspective is considered in facility design. But the plant itself, even though we'll run clinical programs in the manufacturing plant, the fit and finish of the plant are designed to meet commercial standards. And the way that we've designed the air handling, for example, is fairly conservative, again, because this is a facility that we could quite easily want to license multiple programs in. Other companies have made mistakes, in my opinion, where they've built it sort of to a clinical grade and regretted it later, had to retrofit the plant, for example, to meet a new standard that they originally didn't conceive. We decided to build a bit more conservatively with segregation of different rooms and design to ensure that that wouldn't be a requirement. And that also includes the selection of our fill finish equipment, which again is intended to meet worldwide standards in terms of design, which is a very strong area of focus right now for regulators.
And can you talk about how you've secured your supply chain? Obviously, we're in a pandemic, which creates working issues, but also raw materials are being directed towards vaccines. So how do you secure things?
Well, I think it is a challenge. I'm not going to sit here and say, "Wow, this is easy." Our supply chain head doesn't have sleepless nights. She certainly does. But we have for critical raw materials, we did an analysis of all of our raw materials years back, identified those that were deemed critical to the process, that there were no analogs that we could find that were like for like changes on those raw materials. And for, let's say, that 10 or 15 critical raw materials, to the extent that we could, we identified alternate suppliers that can supply materials. We qualified those materials as we did the original ones we were working with, so we established dual supply. And for the few that remain that we can't do that with, for example, we use an inventory strategy to try to stay ahead of that, knowing we have a 2.5 to 3 year forecast that we keep in our plans and that we share with all of our manufacturing network. And we have a raw material strategy. We just implemented an ERP system as well to make sure we stay above preset inventory levels in that case. I think some of the other things that I'll put in with the bioreactor process that we've gone to, we eliminated things like serum from our media. So those are components that you really would worry about access to, and we purposely have eliminated those from our process.
Got it. Now, you've likely been talking about the answer to this all along, but let's try to pull it all together. But what have you learned from the companies that have faced production issues in the past in the AAV space? Just a quick summary, if you don't mind.
Sure. I think our main focus early on was to develop the highest level of product understanding that we could. We have a significant analytical chemistry group within our CMC team, and a significant early investment in process development. And our philosophy from the beginning was to understand our process and understand our product in great depth. That way, no matter where we run this process, whether it's at a CMO, whether it's now in our new manufacturing facility, the highest possible level of understanding of that is available to us because of all the thorough work our CMC team has done.
Good. And then would you touch on points of external validation, GMP certification, et cetera?
Well, we haven't been inspected yet, nor have our contract manufacturing organizations for programs that we're associated with. So right now, our validation is largely in our IND filings, the give and take that we have with regulators, which has been, my view, very successful over the years for multiple programs. And I think more importantly, the seasoned team that we have on board that is very adept at anticipating future needs. So we know when we need to do analytical validation, we know when we need to do process validation, and we know if our processes are performing acceptably and would be favorably reviewed in a future audit. So we have a very strong internal audit program that guarantees we're paying attention to those key pieces. One last thing I would throw in is we've spent an extensive time internally on potency assays. So you've seen a lot of cases where those have been based on mouse models all the way through licensure of programs. Very, very hard to control that type of an analytical method. And what we've done is we've got cell-based potency assays, which are either qualified or validated very early on in our programs to stay ahead of that. So that's a good example I would give of an area where we felt there was risk in the industry, and we've addressed that by proactive, and Ken Mills has been really supportive because that type of work isn't free. It's an upfront investment you make, but you're going to reap the benefits of it down the road.
Got it. And Curran, would you discuss any regulatory trends you're seeing with regards to what you need to do going forward?
There's a lot going on. I think obviously on the FDA side, they see everything. They see all of us, performing in different ways, and they're trying to put all the pieces together as to what is it about CMC that drives safety? What is it about CMC that drives efficacy? And, we see what we see, but they see what every company is doing and are giving us advice one way or the other about how to make the safest products possible. There isn't, there's a lot of guidance on how to develop drugs in this area, but as I said, it's an emerging field. So we're encouraging as much as we can open dialogue with FDA. We want them to come and visit our site because we're located, 15 miles from headquarters, and have exchanges of technology information, our progress. And of course, we have to honor the fact that this is a super busy group right now. So getting their time is difficult. But when this eases, which it will, we're looking forward to, I'd say, more cooperation and back and forth, bringing FDA into what we're doing and making that constructive dialogue, more so than we can in this current environment.
Got it. Now, one question I've asked at this summit that's elicited a variety of responses is to what extent can manufacturing affect systemic tox? So on one end of the spectrum, it's about the capsid. On the other, manufacturing can have a significant impact. What's your perspective?
I mean, I certainly think there is pressure on the concept of what's the total viral load being delivered to a patient, which translates into what's the purity of your product profile in terms of full capsid. So if you believe that the total viral load is associated with safety outcomes, and I think that's very much an open question. I don't think that answer exists yet. Then the only way to enhance your ability to dose at higher levels is to have a higher level of purity related to full capsid. So I think that ultimately, though, I'm a little bit old school in this case, that your toxicology data that's performed in the early stages of your IND, your pre-IND work, as it's associated with your process, your tox lot, that should stand on its own merits. That's why you're doing all of that work originally, realizing there are flaws in toxicology models. They're not 100% predictive. But I feel like that has to be considered, that if you see good safety in preclinical development, it should lead to good safety in clinical development. But that's why we do this long process that we have to ensure all of those happen in concert.
Got it. And then from your discussions with folks like me being in the financial community, just to be clear, what do you think that we miss most about what you do? What's the biggest misunderstanding by analysts?
I think there can be maybe over-reliance on capacity of an organization. How many bioreactors do they have, and at what scale are those? And does this mean they can meet their objective in terms of patients served? And as I described in that supply chain side, I would also be looking at, can they do everything else? Because when you look at situations where people have had clinical holds applied, what have you, it almost never has to do with bulk production. It's always something else. And so I would say just thinking about evaluating companies more on the broad context of their performance than just how much they have in terms of bioreactor capacity would be an important aspect to consider as to whether they can stay on track with their timelines.
Understood. Thank you, Curran.
Thank you.
We now conclude this session.